Bismillahirrahmanirrahim. This is 5090 O-Level Biology. Today we're going to be starting the chapter 4, which is a new chapter which has been added in the new syllabus, and this is called Biological Molecules. Now, as always, I always start with the syllabus. The syllabus is lists the chemical elements that make up carbohydrates, lipids, proteins, and DNA. Then number two state that large molecules are made from smaller molecules limited to starch, cellulose, and glycogen, from glucose, and proteins from amino acids, and lipids from fatty acids and glycerol, and DNA from nucleotides. Now this is new in the syllabus. And then it says part 3, describe and be able to do chemical tests for starch, for glucose and maltose, which is a reducing sugar test or the Benedict solution test, and a protein test, which is also called the Biuret test, and the lipids, which is also called the ethanol emulsion test. So this is all what is part of the chapter 4 syllabus. The first point of the syllabus is the new thing which has been added is the elements. Of course, we had that in the last syllabus as well, but in the new syllabus, this is now being mentioned. So if you look at all of these, there's something which is common to all the 4 biological molecules. Carbohydrates, fats, or lipids, and proteins and nucleic acids, they all have carbon, hydrogen, oxygen. So the 4 of them have carbon, hydrogen, oxygen. Now the only difference is that carbohydrates and lipids have carbon, hydrogen, oxygen. So that is a common thing. But proteins, then we come across, we have a little problem with the proteins. Proteins have nitrogen and sometimes sulfur. Insert 2 amino acids, actually you have sulfur. So there is nitrogen and there is sulfur in proteins. But sulfur is very little, but mainly it's nitrogen. Now in nucleic acids, there is again carbon, hydrogen, oxygen, nitrogen, but now there is phosphorus or phosphate. So phosphates are present here, sulfur is present. So nucleic acids will have PONCH, and this will be, proteins will have CHONS. So this is what we have got to remember, but carbon, hydrogen, oxygen are present in all 4 of the macromolecules. So these are the organic macromolecules. Now let's look at the 4 types of macromolecules. Number 1, we have carbohydrates. Number 2, we have proteins. Number 3, we have lipids. And number 4, we have nucleic acids. Now basically carbohydrates and lipids store energy. Proteins are usually structural components and we have enzymes. Nucleic acids are genetic material which includes DNA and RNA. Now let's come to the first one, which is carbohydrates. Now carbohydrates are made up of repeating monomers, and those monomers are basically glucose units joined together. So glucose is the monomer, and so one glucose unit is called a monosaccharide, and then of course we have disaccharide. So if you have 2 glucose units joined together, this will be a disaccharide, and this will be called maltose. So we have monosaccharides, disaccharides, and then we have polysaccharides. Now you can see the polysaccharides. The first one is starch, and you know starch is a storage molecule found in plants. So you see all these branching, which makes it very compact, and many glucose units can be stored in it. It's like a 5,000 rupee note in which many glucose units are joined together. Similarly, glycogen, which is found in animal cells, in the liver and the muscle cells, that is again a storage molecule, and that is again many glucose units joined together in in chains and in branching patterns. And the third polysaccharide is cellulose, and you can see it's got these. Why? Because cellulose makes up the cell wall, and we need strength. It's not a storage molecule; it's a structural polysaccharide. So you need strength in it, and for that strength, it is all in fibers, and then is joined together to make a thick rope-like structure, so that it can support the cell, because the cell wall should not break. It should not burst when too much water fills in. So the three polysaccharides are starch, storage molecule, glycogen, storage molecule in animals, liver and muscle, starch in plants. Cellulose makes up the cell wall and is a structural polysaccharide. Then disaccharide example, sucrose is a disaccharide. It is made up of glucose and fructose. Lactose is made up of galactose and glucose. Maltose is made up of two glucose units joined together, so it is G plus G. But lactose is made of galactose and glucose. So these are few examples of disaccharides. Now you can see how two glucose units will be joined together and a water molecule exits, and this is called condensation. This is a condensation reaction because water exits and it condenses. But if you want to break this bond is called a glycosidic linkage. But if you want to break this bond, then we'll have to add water. And when we will have to add water, that is called hydrolysis. In the digestion of starch, when you study this in human nutrition, we will study how starch is digested to maltose and then to glucose. So hydrolysis and condensation. When water exits, it's called condensation. When water is added, it's called hydrolysis. Next, we come to the molecule protein, macromolecule protein. The monomer is amino acids, but then we have 20 different amino acids. Now, how are they different? Because in carbohydrates, I told you only glucose. But here now I'm saying they're made up of amino acids, but there are 20 different amino acids. Now, this is what one amino acid looks like. It has 5 basic parts. A central carbon atom, which is this, an amino group, which is this, NH2, an H, which is this, a carboxyl group, which is COOH, this one here. Please pause the video here and have a look at it. And an R group. R group means it is the residual group or it is the variable group. There's no such thing as R. But this is the one which is variable, which can change. Like if I said I have 5 markers, so for what do you say? What is the difference? There are 5 colors, 5 different caps. So this the rest of the structure, which I'm coloring all in black, this is the same in every amino acid. It's only this which is different. And every amino acid, and that is why we have 20 different amino acids. Because the R group is the one which changes, which is variable. Let's try to understand how two amino acids join together and form a peptide bond. So this is a peptide bond formation. So you can see water as exiting, and this is the peptide bond between the carbon and the nitrogen. So this is a peptide bond, and this would be a dipeptide. Why? Because there are 2 amino acids in it, just like we had disaccharide. So dipeptide would have one peptide bond between them, and this water molecule exits. So one amino acid forms with another amino acid, and we have this water molecule exiting and forming a dipeptide. 2 amino acids joined together by a peptide bond. This is called a peptide bond. At times I give you a little more information because I feel you know it, then you can understand it better. But it's very badly given in the book. I don't very like it very much. So I'm giving you a slightly more information than what is required in the syllabus. Now you can see here how the R groups are different. Now you can see glycine is the smallest amino acid. It is only in the R group there's only hydrogen. In alanine it is CH3. In valine we have a whole lot of structure to it. Then in leucine, and please remember we just use the first 3 letters. Like in glycine we just say GLY, gly. And that's how we represent amino acids. Isoleucine is ILE, then methionine is MET, and phenylalanine is PHE, and tryptophan TRP. Now you don't have to know all these names, but you realize how these give different properties. So these are non-polar side chains, these are polar side chains, and electrically charged side chains. These are acetic, these are basic. So basically the 20 different amino acids are because of the different R groups. And these names, some of these names have been mentioned in your book, and I want you to be familiar with these names. So now how will a protein be formed? So maybe the gly is the first amino acid, this one. Then the second amino acid could be anything else, could be met. Then the next amino acid could be gly again. Then the next amino acid could be maybe LYS. So this is the structure of a protein is dependent on the sequence of amino acids in a protein. So the sequence of amino acids in pepsin would be different. The sequence of amino acids in amylase would be different. And that is the primary structure of a protein, how the amino acids join up to form a protein molecule. And surprisingly, this sequence of amino acids is dictated by the DNA. The DNA dictates that number one will be this amino acid, number two will be this amino acid, number three will be this, and number four will be this. So the DNA dictates, so DNA is a big dictator. So DNA dictates the sequence of amino acids in a protein molecule. Now coming to another molecule, lipids, oils, triglyceride or fat. What is it made up of? It is made up of one glycerol molecule. This is not glycerine, glycerine. It is glycerol. Please get the spelling right. And then it has three fatty acid chains. So you can see 1, 2, 3. So three fatty acid chains. That is why when you study this in digestion, the enzyme lipase breaks it up into one glycerol and three fatty acids. Right? So all fats are made up of, so they're not polymers, but it's a macromolecule. So they're made up of one glycerol and three fatty acid chains. Now, we can call them lipids, we can call them oils, we can call them triglycerides. Now, a typical formula for glucose, as you all remember, is what? Is C6H12O6. But a typical formula for a fat molecule is surprisingly C18H34O2. This is just one. Now you see how there is less oxygen in it. But this is more energy rich. Like for instance, if you have 1 gram of fat, it gets you more energy than 1 gram of carbohydrate of a potato, boiled potato or roti. So three fatty acid chains, one glycerol, and less oxygen in it. This is the structure of a fat molecule, which fats are used in the cell membrane. Fats is an energy store. So lots of uses of fats in the body. Next we come to the macromolecule DNA. And what is the monomer? Like we say, this wall is made up of bricks. So what is DNA made up of? It is made up of nucleotides. Just like we said, proteins are made up of amino acids. So DNA is made up of nucleotides, and there are four different types of nucleotides. Now let's look at the structure of one nucleotide. As I told you earlier, there's carbon, hydrogen, oxygen, nitrogen, and phosphorus or phosphate in it. So it is made up of three things. One is a sugar, which is a deoxyribose. So this is a pentose sugar. And then there is a base, which can be adenine, which can be adenine, which can be thymine, which can be cytosine, which can be guanine. And there is a phosphate. So these are the three parts of a nucleotide. There is a deoxyribose sugar, a base, which is the variable one. It can be adenine, can be thymine, can be cytosine, can be guanine. This is the one which can change. The rest cannot change. The deoxyribose will always be there, and the phosphate will also be there. Now here you can see how this nucleotide joins with another nucleotide. and it joins by a phosphodiester bond which is formed between one nucleotide and the other. It forms this single row of nucleotides joined together. Another showing you phosphate, the pentose sugar, which is deoxyribose. In DNA, it is deoxyribose. I'm not going to talk about RNA here. And this is that organic base which can be adenine, thymine, guanine, or cytosine. The four variations to it. Now, this is a very nice diagram showing you the single strand and then the double strand. So, adenine will always pair with thymine, and guanine will always pair with cytosine. Again, if you have thymine this side, you'll have to have adenine this side. If you have adenine this side, you'll have thymine on this side, and there are two hydrogen bonds between it. Similarly, between guanine and cytosine, there are always three hydrogen bonds. Remember, C is the third letter of the alphabet, so three hydrogen bonds between it. So if you have G here, you're going to have C here. So if you have C here, then you'll have G here, and there'll be three hydrogen bonds. If you have A here, you'll have T here. If you have T on this side, then it will be A on this side. If you have G on this side, it will be C on this side. So this is called the base pairing rule. And this is the structure of DNA, which is a double helix. Another diagram showing you the DNA consists of two strips made of sugars and phosphate twisted around each other and connected by nitrogen bases. Looks like a spiral staircase or a twisted ladder. Another diagram showing you the anti-parallel strands. You see, this is directed like this, these are directed like this. This is slightly more information than the O levels, but I still want you to know it. Some of you like this information, some of you want the very basics. And then you see this is called the sugar phosphate backbone, and the bases are jutting in between and forms the ladder. So double helix DNA ladder, anti-parallel DNA strands, and you can see the adenine, thymine, guanine, and cytosine, which have been labeled here. So please pause the video here and have a look at it. Now coming to the food tests, if we test any food, could it be an apple, could be a potato, could be a piece of bread, could be a piece of a biscuit. So you crush the sample because you need to release the cell contents, and then you add iodine solution. Now iodine is brown in color. You can see this color here, iodine. And if the color remains brown when you add it to any food, then that means no starch is present. But if the color changes from brown to blue-black, then starch is present. Now here, please remember, there's no heating involved in anything. You don't have to heat any solution. So please do not write that in the exam, heat the solution or write something like that. So the first test is the starch test. The next test that we have to study is the protein test, which is also called the Biuret test. In this, what we do is we add sodium hydroxide. So we take the sample, whether it's a potato or an apple, and we crush the food sample. And then we add sodium hydroxide. Now this is like water, so we call it a clear solution. You don't say transparent, you don't say see-through, you don't say translucent, you just say it's a clear solution. That's the biological term that we use. So it's just like water. So you add sodium hydroxide, and then you add copper sulfate, which is a blue color. So you add the copper sulfate. So you add sodium hydroxide, and then you add the copper sulfate. And if you find that the blue remains blue, I don't say no change, I don't agree with that. So if the color remains blue to blue, then no protein is present. But if the color changes from blue to purple, then protein is present. So P purple, P protein, P present. So this is a very easy way to remember it. Now another thing which you've got to understand is that the copper forms a complex with a peptide bond. And more the protein, more intense the color. So more dark purple, more protein present. Light purple, less protein present. So this is what you have to understand about the protein test. Again, another very important thing to note is that there is no heating involved in this. I did not say heating anything. So there was no heating in the starch test, and there's no heating in the protein test either. Now coming to the third test, this is called the ethanol emulsion test for lipids. Uh, in this test, you've got to remember is that fats or lipids dissolve in ethanol. Look at the word I've used, dissolve in ethanol. So you take the food sample, you crush it, and you add ethanol. And then when you add ethanol, and if any fat is present, it will give you a clear solution. Why? Because you see, any fat present will dissolve in the ethanol, just like I say, sugar dissolves in water. So if I give you a glass of water, unless you taste it, you can't tell there's sugar in it. Or if I say salt dissolves in water. So you add ethanol, and if there's any fat in the sample, well, it's going to give you a clear solution. You can see this clear solution here. But then when you add water, it gives you a cloudy white emulsion, and that shows fat is present. But if it remained clear, then no fat is present. Now look at this procedure that I've given you here. Add 2 centimeter fat or oil to a test tube containing 2 centimeter cube of absolute ethanol. Dissolve the lipid by shaking vigorously. Add an equal amount of cold water. Observation: a cloudy white emulsion. Naturally, if fat is present, but if it remained clear, that meant fat wasn't present. Now the basis of the test is lipids are immiscible with water. Adding water to a solution of the lipid in alcohol results in emulsion of tiny droplets in the water which reflect light and give a white opalescent appearance. So this is the basis of it. Why does it become cloudy white? This is the main reason why you've got to understand is fat will dissolve in the ethanol, but when you add water, then the lipid forms these tiny droplets in the water. Just like if you've had something with a lot of butter in it, like if you've had a paratha with a lot of ghee in it, then when you wash your hands, the water and the fat doesn't mix. And unless you use soap, only then you can wash away the ghee or the butter which was in the paratha which you had. Now we're going to discuss the last test, which is the Benedict's test, which is also called the reducing sugar test. Now in this, what we need to do is we need to take 2 centimeter of the glucose solution, and we needed to add Benedict solution to it. So 2 centimeter Benedicts and 2 centimeter of glucose solution. And then you heat in a water bath. The temperature of the water bath should be anything between 80 degrees Celsius to 100 degrees Celsius. So if you said boiling water bath, that would be also fine because water boils at 100 degrees Celsius. And then you see the color change. If the color remained blue, then it means there is no reducing sugar in it. So no reducing sugar. This is reducing sugar. No reducing sugar present. But if it turned to a brick red precipitate, that means reducing sugar present. Now, this is also called a semi-quantitative test because it roughly tells you the quantity of reducing sugar in it. So if it remains blue, no reducing sugar. If it's green or yellow, then it traces of reducing sugar. Orange, moderate, and brick red precipitate means large amount of reducing sugar present in it. So that is why this is called a semi-quantitative test. Semi means half and quantity means how much there is in it. So it roughly tells you the quantity. So if there was one solution which was orange and one which was brick red, so you knew that this one had less reducing sugar and this one had more reducing sugar in it. So you roughly get to know less or more, but not the exact amount. And last, a quick summary of all this testing. Benedict's test is for reducing sugars. Iodine test is for starch. Emulsion test is for lipids, and Biuret test is for proteins. Now, please remember all these three, there is no heating. No heating at all. Please do not write. This one, the only one in which there is heating is involved is the Benedict's test for reducing sugar, in which we have to heat it at 100 degrees Celsius in a water bath. So this is, you can see this is the fat test, this is the negative Biuret, positive Biuret, and a positive starch and a negative result for. So, and these of course are the different reducing sugars, no reducing sugar, traces, low, moderate, and high. So this is just a quick recap of all the tests, and the important thing is that which ones do we heat and which ones we don't heat. So the Benedict's test we heat, the others we do not heat. That completes this chapter, and thank you for watching, and thank you for subscribing. This completes chapter 4, and there will be no more videos on it. This chapter was a short chapter, so there's only one video on this chapter. Thank you very much.